Afternoon, ladies and gentlemen. Thanks for joining GenScript webinar today. I'm your host, Boon Hua. Today, we are going to discuss how to enhance your Western blot workflow with advanced technology. GenScript is a global leading biotech company providing life science services and products. With reliable spreadsheet service like gene and peptide synthesis, protein expressions, antibody productions, protein analysis solutions, we are able to help scientists and researchers to enhance your research project in a more productive and effective manner. Our speaker today, Dr. Jen Tan, is currently a product manager of catalog products with GenScript. Dr. Tan graduated with a PhD and worked as a research fellow at the Singapore Institute of Clinical Science. Her research area focused largely on establishing an epigenome for clinical samples and involved collaborations with government hospitals. So before we get started, here are some tips for you. On your screen, you are able to see a Q&A box. Resources that help you better understand our offerings. And on the bottom right, there's a survey questions to help us better understand your need. And if you need any demo sessions with us, please feel free to hit the request demo button in the middle of your screen. So without further ado, I'll pass the stage to Dr. Tan.
Okay, thank you so much, Boon Hua, for the very kind introduction. Hi, good afternoon, everybody. Thank you so much for joining me here today. Okay, so basically, I am Jane, and I'm the catalog product manager from GenScript. Okay, so for today's webinar, we will be talking about how you can actually enhance your Western blot workflow with some of the advanced technologies coming from GenScript itself. So for those of you who are currently performing a Western blot or are thinking about performing Western blot, do consider some of the solutions that we actually have at GenScript, because the solutions that we have can actually help you simplify your entire Western blot workflow. Okay, so that's what we will be talking about for today. But before we actually talk about the solutions that we actually have, firstly, let's go through some of the basics of Western blot. Okay, so firstly, why is Western blot performed? So Western blot is actually a very powerful technique that you can actually use to help you detect specific protein molecules in a sample. Okay, so usually your sample likely contains not just one protein, but many proteins. Okay, so it's usually a mixture of proteins. And sometimes you just want to detect and see whether or not your sample actually contains a specific protein of interest. So you can actually perform Western blot to give you to get you your answer. Okay, not just that, it can also be used to estimate the size of your protein of interest and also its relative expression levels. Okay, so for example, if you look at this blot result over here, okay, so over here you have 8 samples in which, as you can see, all 8 of them actually contain that protein of interest because all of them have that same band over here. Okay, so all 8 samples contain that protein, but then what other information can you actually get out of a Western blot? So like I mentioned, if you were to actually run a protein ladder like this along with your samples, then what you get additionally would be the estimated sizes of the protein of interest. Okay, so as you can see in this case, according to this ladder, your estimated size of your protein is approximately 50 kilodaltons in size. Okay. And not just that, you can also compare its relative expression levels from sample to sample. So for example, you can see over here, you have 8 samples. And as you can see, the intensity of the bands are a little bit different from sample to sample. So for example, in sample number 4, you can see it's slightly darker in terms of band compared to sample 5. So with everything else normalized, you can actually, tell that maybe sample 4 may contain a little bit more of this protein compared to sample number 5. So these are just some of the information that you are able to get out of a Western blot. Okay, so it sounds very simple, right? In analyzing a Western blot result, it's relatively easy to analyze, but when it comes to actually performing the Western blot itself, there are actually quite many steps that the end user will have to go through before you can actually interpret your results. Okay, so some of these steps in Western blot would be things like sample preparation. Okay, so you need to prepare your samples first before you actually load them onto your gels for gel electrophoresis. Okay, so gel electrophoresis is extremely important because this will definitely affect the way your proteins are finally being separated. Okay, so after gel electrophoresis, then the end user can choose to actually perform a gel staining or a gel to membrane transfer. Okay, so this is up to you to decide whether you want to first perform a staining on the gel before you perform that transfer. And then once you've performed that transfer and you've transferred all your proteins from the gel to the membrane, then finally you perform that blocking step as well as antibody incubation. And this can then help you finally visualize your proteins on the membrane. Okay, so these are all the different steps that you need to go through just to actually get your final Western blot results. And in terms of the solutions that we have at GenScript, okay, I would say we do have quite a wide range of solutions, starting from sample preparation all the way to blocking and antibody incubation. Okay, so this is what we will be going through for today. But before we actually talk about the solutions, first let's talk about gel electrophoresis first. Okay, so gel electrophoresis is again extremely important. So what happens during electrophoresis is basically protein separation. Okay, so again, you start off with a sample, and this sample likely contains many different types of proteins. So remember, the reason why you want to perform this electrophoresis is because you want to separate out your proteins in your sample first, okay? And usually it's via differences in molecular weight. And why do you want to separate out your proteins of interest? Because remember, your samples contain a mixture of proteins. Okay, so in order for you to more accurately detect your protein of interest, you need to first separate them out, okay, before you can more efficiently perform that detection later on. So this is what protein separation or electrophoresis is for. Okay, so you want to separate out your protein. So how do you actually perform electrophoresis? It will be in this gel tank, this setup like this. Okay, so you have a gel tank in which in the middle, you can see here, there is actually a gel. Okay, so your gel is actually in the middle of two plates like this. Okay, so your gel is inside. And your gel is typically made up of two portions. The first one is a stacking gel, which is on top. The second portion is a running gel, or some people call it the resolving gel. Okay, but these two portions make up your actual gel. Okay, I won't really go into the details of each of these portions. But so this is how your gel looks like. Okay, and at the top of the gel, you have grooves like this, which is basically your wells. So this is where you would actually put your protein sample, okay, into each well. Okay, and then what happens is that you have your negative electrodes on top and positive electrode towards the bottom. So what happens is that once you have pre-treated your samples, okay, usually with maybe SDS or LDS, some sort of a loading buffer, pre-treat your samples. So this pre-treatment process usually ends up with your proteins being negatively charged. Okay, so your proteins are loaded here, negatively charged. And once you actually turn on the voltage across this pair of electrodes, what happens is that your negatively charged proteins will then move downwards, towards the bottom of the gel, because towards the positive end of the electrode. Okay, so your proteins all start to move downwards, and once it actually hits the running gel, so that's when they actually start to separate from each other via differences in their molecular weights. Okay, because there is a certain pore size associated with your gel, and the larger proteins will then move more slowly across the gel, okay, because they are more impeded in terms of movement, whereas the smaller proteins will then migrate much more quickly across the gel. Okay, and therefore they are separated based on the differences in terms of their molecular weights. Okay, so this is what happens during electrophoresis. Okay, and so basically, as you can see, right, in electrophoresis, there are so many parameters that you can actually play with. Okay, so all these things that I just spoke about, basically, how do you perform that sample pre-treatment? Okay, how are your proteins treated before you load it onto the well? What sort of gel are you using on this gel tank? Okay, are you using a tris glycine gel? Are you using a bis tris gel, and so on? What running buffer are you using over here? Because as you can see outside the gel, you have your running buffer all over in the tank. So all these things will all affect how your final protein separation actually looks like. So all these parameters together, they work together to actually give you your final separation outcome. Okay, so the way your proteins are separated, all these factors will play a part. And of course, even the voltage that you use, how long you actually perform your run, all these factors will play a part in how your final protein are being separated. Okay, so again, very, very important, but I won't go through all of the details over here or all of these parameters, okay, but just again, do make sure that you understand that all these things together work together in terms of giving you your final protein separation. Okay, but what is really important is the type of gel that you actually use. Okay, so I'm not too sure whether most of you are actually aware that there are actually different types of gels available, okay, in the market. So different types of gels are actually made using different buffer systems. Okay, and therefore the different gels are actually maintained at different pH values, and this actually is important when it comes to protein separation, because you know proteins are extremely sensitive when it comes to a change in pH. So you always want to make sure that your proteins are stable when you actually run your actual gel electrophoresis. So the choice of gels that you use is extremely important. Okay, so I would say there are many different types of gels, but over here I've mainly only listed two main types. Okay, so the tris glycine gel, which is, I would say, the more traditional type of gel that most people are used to using. Okay, and then you've got also bis tris type of gels over here. So if you look at this table, which I basically just very quickly summarized some of the differences between the two types. So tris glycine gel, although it's, I would say, the more conventional type of gel that people tend to use, as you can see, in terms of its pH, it's slightly more alkaline in pH, okay? So its pH is about 8.8. So this could potentially actually cause the hydrolysis of your polyacrylamide gel. So this actually affects the stability of the gel itself. Whereas for bis tris type of gel, it's slightly acidic to neutral in terms of pH. So this actually increases the stability of the gel itself. Okay, and not just that, it also suppresses cysteine reoxidation, and this is important because you do not want your denatured proteins to actually reoxidize back. Okay, so this is great, basically, bis tris gels, because they are a lot more stable, not just for the gels, but also for the protein. Another observation that has actually been made is that for tris glycine gels, it is found that when you actually run tris glycine gels in terms of the electrophoresis run, right, the operating pH actually reaches about 9.5, which is pretty high. Okay, and this may actually distort proteins. So again, tris glycine gels may not be that great in terms of the stability of proteins, whereas for bis tris gels, in terms of the operating pH, it's around neutral when you actually perform your electrophoresis run. So this actually improves protein stability. Okay, so all in all, right, you can see that bis tris gels are generally just more stable compared to tris glycine gels. And the impact of that is that you tend to get better looking bands with better separation coming from bis tris gels compared to tris glycine gels. So if you find that your separation isn't that great, you do see fuzzy bands that are not that sharp, you could try bis tris type of gel because it can be a lot more stable, not just the gel itself, but also for the proteins that you are running on the gel. Okay, so just do take note that there are different types of gels available. Okay, and what is the differences in terms of their stability. And not just that, of course, you can also select the different percentage of polymerization. So if you look at this chart on the left hand side, you can see that for a particular chemistry of gel, you can choose the percent of polymerization over here. Okay, so depending on your target protein that you are looking at, let's say, for example, if I wish to separate my proteins at maybe 20 to 30 kilodalton range, then you can actually refer to a chart like this, a reference chart. For example, we have this chart at GenScript. So you can actually see and take a look, approximately what kind of percent gel would actually give you the best separation outcome for this range of molecular size of proteins. So let's say if I were, to have like a 20 or 25 kilodalton protein that I wish to detect, then probably a 12% gel actually works really well in trying to separate out these proteins. Compared to, let's say, an 8% gel, you can see that it's not giving you very, very good separation between 20 and 30 kilodaltons. So you can actually refer to charts like this to give you an idea of what percent gel should you use for your particular protein that you're interested in separating out. Okay. So not just that, but of course, remember that when you run these gels, okay, you have to run it with a certain running buffer as well. And there are certain running buffers that have been also optimized to actually be used with different types of gels as well. Okay, so all those factors that I mentioned, whether it's a running buffer, your sample loading buffer, your type of gel that you are using, the voltage that you are using, all that plays a part in terms of giving you your final protein separation. Okay, but again, it's always good to understand that there are different gel types available. So this then leads me to actually introduce GenScript's precast gels that we actually have. So it's actually called the SurePage precast gels. Okay, so it looks something like this. And then, of course, our gels that we have are bistris in terms of chemistry. So if you recall from the previous slide, bistris gels tend to be a little bit more stable compared to tris glycine gel. So our GenScript's SurePage bistris gels are more stable in that sense. Okay, and so these are just some of the features and benefits of our SurePage precast gels. So the first thing, like I mentioned, it is bistris type, okay, in terms of chemistry. So pH is somewhere towards neutral. So this is more stable for proteins, okay? And therefore, the benefits that the end user tends to get would be you get clearer, sharper bends, and it's definitely going to be better in terms of performing that transfer, detection, and quantification. Okay, so in terms of your actual bands that you see, you tend to get much clearer, sharper bands and better resolution. Okay, and not just that, we've also tested our SurePage gels, and we find that they are actually extremely good to be run at about 180 to 200 volts. So this means that you can actually run it pretty quickly. So within 20 to 30 minutes, you should be done with your run, typically. Okay, so it's pretty quick in terms of gel separations. So you can get that higher throughput if you need, because you can actually run it at about 200 voltage. Not just that, if you were to look at the welds, okay, on top here, if you look at GenScript's weld design, you can see it's slightly different from the regular weld design over here. Okay, so our welds are a little bit larger in terms of volume. Okay, so you can actually load, I believe, at maximum 80, but probably around 70, 60 to 70 microliters of samples into each weld. Okay, so this actually, the benefits of this is that you can load more samples into each weld. Okay, and also there's less spillage across, towards the next subsequent well. So this means that there is a higher chance of you detecting low concentration proteins. So if you have certain samples in which your protein of interest is present at very, very low concentrations, then having a larger well volume will be useful, because you can actually load more into each well, and hopefully you're able to then detect your protein of interest. Okay. And of course, also less well-to-well contamination, because it doesn't spill over that easily. Okay, and the SurePage gels that we have are also compatible with many common gel tanks. So you do not need to really purchase a new gel tank just to use our SurePage gels. So even though, if you have another vendor's gel tank currently, you can actually use that and run it on our SurePage gels as well. So it's very convenient. And one important factor is that if you purchase our precast SurePage precast gels, we actually provide free running buffer to you as well. So we actually will ship a free running buffer along with your purchase. So in that sense, it's going to be very convenient for you and also very cost savings. Okay. And the shelf life actually of our precast gels is about 2 years if you keep it in the fridge. Okay, so this is pretty long shelf life, I would say. Okay, so you can actually store them and you do not need to worry about the fast expiry because you can store them for about 2 years. And it's extremely price competitive. So do reach out to us if you're interested in actually trying them out. I would say it is really very, very cost competitive. Okay. And of course, the fact that it's a precast gel, so this means that the advantage is that you do not need to self-cast these gels, especially if you are working with gradient gels, which can be a little bit more troublesome to cast. And it can be therefore very time-saving. And not just that, it gives you more uniform results, okay, and consistent results as well. And you do not need to work with toxic reagents like acrylamide. Okay, so these are all some of the features and benefits that you can get out of using our SurePage gels at GenScript. So this is just some of the runs that we've done internally to compare our SurePage Bistris gels compared to other types of tris glycine gel, whether it's precast or homemade gels. You can just generally see that the bands that you get with the SurePage gels are a lot sharper compared to tris glycine gels. So tris glycine gels tend to give a little bit more fuzziness in terms of the results compared to Bistris gels. So if you're getting fuzzy bands or bands that do not really resolve very well, you can actually give the SurePage Bistris gels a go. See whether that actually helps you improve on your resolution. So this is actually just some of the data that we actually got from customers when we actually did a demo at a customer site. So this is actually some other brand of gel over here. Okay, so you can see that the customers actually ran their own samples, and you can see that somewhat the bands, the area in between bands, it's not very clean. You can still see some fuzziness around the bands as well. So not very good resolution obtained over here. But then this was on the SurePage gel. You can see very clean bands observed. Okay, so this is the sort of difference that you may observe when you are using Bistris type of gel compared to other tris glycine types of gels. Okay, so again, do feel free to reach out to us if you wish to actually give the SurePage gels a try. And not just that, like I mentioned, SurePage gels have higher sample loading volumes. Okay, so again, what this means is that you tend to get less of this sort of spillover onto the next lane. Okay, so you get less contamination. So if you look at these two companies of gels, okay, so company B has a lower loading capacity. So this means that it actually overfills very quickly. To load 10 microliters, it's still okay. But as I increase the volume of my samples that I load into the wells, you can see that it starts to form. You can see some spillage across onto the next lane. And as I input more samples, the spillage gets even more drastic. Okay, so having a larger well volume like this, you can see that it actually helps with minimizing that spillage across to the next lane. Okay, and not just that, of course, it also allows for detection of low abundant proteins as well. Okay, so this is the benefits of having a larger well volume. Okay, so just to summarize the SurePage gels that we have, again, in terms of chemistry, it's a Bistris type of gel. And so this is great for gel stability as well as protein stability. So what you get will be very clear bands, very sharp bands with improved resolution. And also the well volumes are slightly larger in size. So this gives you less spillover and improved detection of low concentration proteins. And the shelf life is also extremely long, about 2 years. Okay, and so this means that you can actually store them for a longer period of time. Fast run times as well, because we've tested our SurePage gels to be able to run at 200 voltage very well. And not just that, again, like I mentioned, if you were to purchase our precast gels, we can actually provide free running buffer to you as well. And these running buffers are compatible with our SurePage precast gels. Okay, so I would say quality-wise, extremely good. Okay, do feel free again to just reach out to us to try it for yourself. We are more than happy for you to actually try out our gel. So just reach out to us for free samples of these gels if you wish to try it. Okay. So apart from these gels, of course, like I mentioned, at GenScript, we actually do also provide all the other consumables that you will require to perform gel electrophoresis. So for example, we have gel tanks like this. We have protein standards that you can run alongside your samples. We also have a running buffer. Like I mentioned, you can purchase this separately if you wish. And we also have sample loading buffer as well. Okay, so all these can be used if you are performing a gel electrophoresis on top of our SurePage gels. Okay, so that's actually electrophoresis. And then the next thing is that the analyst or the end user, like yourself, can then proceed to perform either a gel staining or protein transfer. So after you actually perform your electrophoresis, likely you will not really be able to see any of your proteins on the gel itself. Okay, so some people, before they perform a protein transfer, they want to first stain the gels first, just to visualize the proteins and to see how are they separated on the gel before they perform the next transfer step. So to perform this protein staining, so people, again, they perform protein staining in order to detect the presence of the proteins on the gels without performing Western blot or before performing Western blot. Okay, so some people, they do not wish to really perform the rest of the Western blot workflow. They just want to perform staining, but some people, they want to perform the staining before they actually perform the rest of the Western blot step. Okay, so in order to perform this protein staining, you can actually perform this manually, okay, using basically just manually performing the staining yourself like this. Or of course, you can also automate the entire process. Okay, so the manual staining is a fairly popular method because all you need is just a container like this containing some staining solution. Usually it's Kumasi blue based. And then what you do is basically once your gels have finished with the protein separation, you can then put your gel and immerse it in this staining solution. And so what happens is that the dye inside this staining solution will then interact non-covalently with proteins. Okay, so it will bind to the proteins on the gel and therefore stains your bands blue. Okay, but at the same time, the gel also will stain your polyacrylamide gel. So what you need to do usually after you perform that staining is that you also need to perform a de-staining step in order for you to remove any background staining on the gel. Okay, so this is you can do this entire process manually like this, or of course you can actually use an automated instrument to help you perform this staining. Okay, so if we look at manual staining methods, okay, there are basically, I think, two main types. Okay, the first one is the conventional method, where you use conventional staining reagents like Kumasi blue to perform this manual staining. Okay, so the conventional method, in terms of the staining time, it takes at least an hour to a few hours, or some people even perform this staining overnight. Okay, so the staining process can be pretty long. And as I mentioned, likely you need to also perform a de-staining step just to actually make sure that you remove the stain from the background of the gel itself. Okay, it can take many hours as well. As you can see, usually a few hours. So overall, the conventional manual staining method can take about many hours to actually perform this actual staining. But then there is also a quicker method, manual method, where you actually use things like instant blue, okay, Kumasi stain, and this can actually help you fasten your stain time to about 15 minutes to an hour, okay? And supposedly you may not need to perform that de-staining step, but I do know some people still perform that de-staining step just to try to remove as much background as possible. Okay, so this is what you will tend to get at the end of your staining. This is how you can actually visualize your proteins on the gel itself using this blue dye. Okay, so this is what you get manually. And of course, if you do not wish to perform this manually, you can consider an automated instrument, which is what we have at GenScript, called the eStain L1 protein staining system. Okay, so it looks like this. It's a pretty small device, not too large. And so this can actually help you automate the entire staining process. So you don't have to do this manually. You don't need to spend that many hours at the bench waiting for this to happen. So one of the key features of the eStain L1 system that we have is that this actually contains our patented protein staining technology here. And the advantages is that it stains and destains proteins all within just 10 minutes. So within 9.5 minutes, you will be done with your entire staining process. Okay, so it actually cuts short the staining time from many hours to just 10 minutes. So this results in a higher throughput than performing it manually. And not just that, we also have a staining kit that you can actually use with this device. Okay, so the staining kit looks this here, and it's also Kumasi blue based. So it's found that actually, in terms of the sensitivity that you get if you were to use the eStain L1 system, it's very similar in sensitivity compared to performing your long manual staining methods. So in that sense, you do not compromise on the results at all. Okay, it would be very similar to what you would get if you were to perform this manually. But of course, you finish this all within just 10 minutes. And it's probably the only automated device currently in the market, which is unique to GenScript. And of course, again, because it's an automated instrument, this helps you improve reproducibility and consistency of staining results. Compared to if you were to do it manually, likely you get more variability when it comes to batch-to-batch staining compared to actually doing it using an automated system. Okay. And one interesting application is that not only can you use this eStain system to stain your gels, you can also use this eStain system to actually perform membrane staining with Ponzo S. So for some of you who actually perform your gel to membrane transfer, you can actually consider also staining your membrane just to view and see, to check, how are your proteins being transferred onto the membrane. Some people actually wish to perform this Ponzo S staining first, and you can actually use our eStain L1 system to perform this Ponzo S staining. Okay, so it's very flexible. You can use it not just for gel staining, but also for membrane staining. Okay, and in terms of the software, it's very, very simple to use, very easy to program and operate. It's really not much user training required. So this is, you don't have to worry about it being too complicated to use. In fact, we actually do have default programs already set in the system. And using those default programs generally gives you very, very good results. So all you need to do is really just to put your gel into the instrument and just press play, and then it should be done. Okay, so it looks something like this. Again, you can see the top here. This is the display, a very simple display screen. The bottom here, there are some keypads that you can actually use to toggle some of the parameters if you need to. But again, like I mentioned, usually the default programs will run fine on most gel stains. And as you can see at the bottom here, you have two different slots, okay, two different channels. And this means that you can actually put two different gels in here at the same time, and you can stain two gels at one go. Okay, so this is what you do. You take out this gel holder and then put your gel in there that you wish to stain, close it back, and then put this holder back into the slot, and then just press to start your program. Okay. And at the left-hand side, you have also staining solutions and destaining solutions that you can actually connect to the instrument. So you do not need to get really down and dirty to doing the actual staining yourself. You can actually just let the instrument take in the solutions and perform that staining for you. Again, all within just 9.5 minutes. Okay. And the end result that you tend to get would be very sensitive results, very, very good staining results, as well as them being very even and reproducible. Okay, so you can see here, this is a gel run where you can see that we've actually ran protein standards on every lane, and then stained them, and you can see that it's stained very, very evenly. So if having reproducible stains is something that's important to you, really having an automated instrument can actually help with minimizing any variability. Okay, and this is just some examples of showing you how we can also use the e-stain, not just for gel staining, but also for membrane staining. So this is actually your membranes, your nitrocellulose membrane and PVDF membrane after performing that gel to membrane transfer. And some people wish to actually observe the proteins on the membrane. So you can actually perform a Ponzo S staining using the e-stain L1 system as well. So we do have application notes on that. And you can see that performing this manually or using the e-stain L1 system, you get pretty similar results. Okay. So just to summarize, GenScript's e-stain L1 system, it's probably the only automated protein staining device in the market right now. And because it being an automated system, this ensures batch-to-batch staining reproducibility. It completes the entire staining and destaining process within 10 minutes. So 10 minutes, you are done with your staining. Okay, very, very fast. This cuts short a lot of the staining time. And it's very easy to set up as well. One button operation. Really, it's a very simple software to use. Okay, and this of course minimizes handling mess because it's an automated workflow. So the end user, you yourself, you don't need to be handling the actual gel, performing the washes and so on. Okay, results that you tend to get will be crisp blue bands with minimum background. Okay, so again, do feel free to reach out to us to request for a free demo because we do have many demo units available. So if you wish to actually see for yourself to see how fast this instrument actually works in terms of staining, do feel free to reach out to any one of us for a demo session, and you can try it out for yourself so you can actually see how fast it actually works. Okay. So this is if you're actually interested in performing a staining of your gel. Okay, and then after you've performed staining of your gel, if you realize that okay, your proteins seem to be separated very well on the gel, next you can then decide to perform the actual rest of the Western Blot workflow. Okay, so the next step that you can actually perform is a gel to membrane transfer. Okay. So when you perform a gel to membrane transfer, the reason why you want to perform this is really because, so imagine your proteins are currently on a gel, okay, a polyacrylamide gel. And polyacrylamide gel, it's not exactly a solid support, okay? So the proteins in there are not really immobilized enough in the gel, okay? So they can still be diffusing within the well itself. Okay, so in order for you to be able to handle these proteins better, it's better to transfer them out of the gel and onto a solid support like a membrane. Okay, and this helps improve handling as well as target accessibility. Okay, and the main purpose is really to just get all the proteins, the exact same protein pattern of the gel and onto the membrane. Okay, so this is the main purpose of a gel to membrane transfer. And once you've actually transferred your proteins onto the membrane, these proteins will bind to the membranes via hydrophobic interactions and so on. Okay, so that's how the proteins actually stick onto your membrane. Okay, so this is the purpose of performing that gel to membrane transfer. And in terms of performing gel to membrane transfer, there are two main traditional methods. The first one is the wet transfer method, and then you've also got the semi-dry. So both of these are very, very popular methods. Okay, so in terms of the differences between the two, if you look at the typical wet transfer method, in terms of the transfer efficiency, tends to be extremely good. Okay, so this is the gold standard in terms of the transfer efficiency. So the wet transfer tends to give you very good transfer efficiencies, regardless of the size of the protein. Okay, but of course, you likely will need to use a lot more transfer buffer to fill in the entire tank when you perform this wet transfer. And in terms of the transfer duration, it can be pretty long as well. So usually an hour or more than that. Okay. In terms of semi-dry, reason why it's also very popular is because in terms of the transfer efficiency, I would say it's pretty good, but it may not be that great for large molecular weight proteins. Okay, so there may be some limitation, especially when it comes to larger molecular weight proteins. Semi-dry transfer systems may not be able to transfer them that efficiently. Okay, so if you do are looking at larger proteins, then this just do take note that there may be a limitation with semi-dry transfer systems. In terms of the transfer buffer that you need to use, it's a lot lesser, and it's limited to the sandwich in this setup. Okay, and in terms of the duration, semi-dry transfer systems tend to be much faster when it comes to the transfer. Okay, so this is really the pros and cons of each method of transfer. So wet transfer, it's great because of the transfer efficiency, but as you can see, it takes a long time to perform this transfer. Semi-dry, on the other hand, may not be really the gold standard when it comes to transfer efficiency, but then it does the transfer very, very quickly. Okay, so each of them, they do kind of have their pros and cons. So what about this eBlot L1 system that we actually have at GenScript that you can use for protein transfer? So I would say eBlot is essentially the best of both worlds that we just about. So eBlot, it is also a patent pending technology that combines the benefits of a wet transfer as well as a semi-dry transfer system. Okay, so all the benefits that we spoke about earlier on of a wet and a semi-dry is all found within the eBlot L1 system. So what do you expect with eBlot is that you get great transfer efficiencies of all molecular weight proteins, just like your traditional wet transfer, and at the same time, fast run times, just like your semi-dry transfer systems, okay? And less transfer buffer used compared to traditional wet transfer systems. So what this means, the benefit to an end user like yourself, is that this increases the chance of detection of all sizes of proteins in your sample. So you do not need to worry if your samples or if your proteins of interest is higher molecular weight. You don't need to worry that this will not transfer well, because this is essentially a wet transfer system, but it's able to perform that transfer in a very fast time. Okay. And of course, you get higher throughput because you tend to get, because as you can see, the runtime is very, very short for this eBlot one. And at the same time, you do not need to use as much buffer as compared to if you were to run it on a typical wet transfer system. Not just that, again, you can see that there are two independent channels that you can use for simultaneous transfer. So if you have two different gels that you wish to perform a transfer, you can do two at the same time. Okay, so this is a slightly higher throughput compared to just having one gel transfer at one time. Okay, and not just that, it's also a very simple software that is very, very easy to use. You do not really need much training for it. Same thing like the eStain. So if you recall the eStain system that we had, it's exactly the same looking instrument, except that the eStain is blue in color and the eBlot here is green in color. Okay, so but in terms of the outlook, you can see that they both look very, very similar. Okay, and the programs or the softwares are very, very easy to use as well. And it is also compatible with different types of gels, so you do not need to worry that you need to use GenScript's gel on this system. You can use whichever gel you have, whether it's homemade or even gels from other brands. You can actually use it on this instrument as well. Okay, so it's very, very flexible. So again, this is how it looks like. You have your display here, and then you have your keypad, and then you have two channels for you to do two transfers simultaneously. And for each gel holder, if you were to take it out, so all you need to do is just to put your sponge, your membrane, your gel in a certain orientation, close it back up, and then just put it into this slot here, and then connect your transfer buffers into this instrument, and then just press run, and then it will complete this run for you. Okay, so typically it takes about 9 to 17 minutes to complete a transfer using the eBlot system. Okay, so it's a lot faster compared to the traditional wet transfer. So if you look at this table, again, to compare again, now I've added the eBlot system. So in terms of transfer efficiency, it's great for all molecular weight proteins. So it's similar to a wet transfer in terms of its transfer efficiency. But in terms of transfer duration, you can see that it takes about 9 to 17 minutes. So it's a lot faster than a traditional wet transfer system. So it's more similar to like a semi-dry when it comes to transfer time. Okay, so this is actually the only wet transfer system in the market that does a fast wet transfer, such that you actually combine the benefits of these two traditional methods in the eBlot. Okay, so this is just some protein transfer comparison that we've done. And you can see that there are three different types of proteins here: EGFR, TAC, and GAPDH. So EGFR is considered quite a large protein. And as you can see, when you actually transfer it using the traditional wet transfer versus device 1 and device 2, which are semi-dry transfer systems, and the eBlot, you can actually see that there is more bands being observed on the eBlot, okay, especially if the band is of low concentration. You can see that the eBlot is able to very, very successfully transfer this very large protein, such that you're able to see your protein even if they are present at very, very low concentrations. Okay, but you may not be able to see it with a semi-dry transfer system. Okay, so this is, again, the benefit of our eBlot. Same thing, even for TAC, you can see that there is actually a much higher transfer efficiency coming from the eBlot as well, compared to device 1 and device 2, and even compared to the wet transfer method. Okay, so again, with the eBlot, you don't really need to worry that transfer efficiency may not be great for higher molecular weight proteins, because it does have the transfer efficiency similar to that of a wet transfer. Okay, so GenScript's eBlot L1 system, it's really the only fast wet transfer system in the market. Completes the transfer within 9 to 17 minutes. And the transfer efficiency is as good as traditional wet transfer systems. It's very easy to operate in terms of the software, higher throughput because of the fast wet transfer. And again, you can transfer your small, medium, and large proteins very quickly and efficiently with the eBlot. Okay, so again, we do have many demo units available. So do feel free to reach out to us if you need a demo session. We will be more than happy to actually show you how the eBlot actually works. In just 17 minutes, you can actually transfer the proteins very, very effectively. Okay. Okay, so that's for protein transfer. So assuming that you've performed your protein transfer on your membrane, again, at that, as of transferring your proteins, you will still not be able to really view your proteins on the membranes yet, right? So at this point, you can actually decide to perform a Ponzo S stain or continue with the rest of the Western Blot workflow. So the Ponzo S stain, remember, like I mentioned, you can use the E-stain to actually stain your transferred proteins on the membrane using the E-stain system. Okay, so you can, after you perform your gel transfer, then you can actually put the to perform a Ponzo S stain. Okay, or of course you can choose to continue on with the rest of the Western Blot workflow. So if you choose to continue on, then the next thing that you want to do is to actually perform that blocking and incubation step. Okay, so you have your membrane here. So the first thing you want to do is to block, because you want to prevent any non-specific binding of your primary and secondary antibody on the membrane. Okay, so you perform that docking first to ensure that your membrane is effectively blocked. And you can use milk or serum and so on for performing this blocking step. Okay, so once you've blocked your membrane, then the next thing you want to add is your primary antibody. So this primary antibody will then bind to your particular protein of interest, okay, like this here. And this will actually recognize and bind to your protein antigen here. And then what you need to do after that is to perform multiple washes after you perform this primary antibody incubation. And this helps to remove any excess unbound reagents and also remove any or reduce any background. And then finally, once you're done with the washes, you can then add your secondary antibody, which will then help you detect this protein on your membrane. Okay, and then again, perform washes before you actually visualize your proteins. Okay, so all these steps are typically done manually. All these blocking, primary antibody incubation, the wash steps, secondary antibody incubation, all these are typically done manually, which can take up a lot of time on the bench. Okay, so if you look at a typical staining or incubation protocol, you can see that all these steps, the blocking part, 1 hour at room temperature, and then the overnight incubation at 4 degrees, washes with TBST, secondary antibody incubation, all these steps typically will have to be done manually on the bench itself. Okay, so apart from doing this manually, if you find that it's actually really too long a time, you could actually consider performing this using an automated instrument. And this is what we have at GenScript for this. Okay, so using the EasyWest light automated Western device, which looks like this. Okay, so this can actually help you automate all those steps that we saw earlier. So all these steps can be automated within this EasyWest device. Okay, so this helps perform that automation of the blocking, the antibody incubation, and the wash steps. So this means that you minimize any manual work, and the same protocol can also be saved over here. So you can just use the same protocol on various batches of samples. Okay, so what this actually does is that it saves you many hours of manual work, and not just that, it also minimizes variability as well. And it's been found that this step of the Western Blot workflow actually causes the most source of variability in the entire Western Blot step. So by being able to automate this step, you can actually help to minimize this variability. So you get more reproducible and more consistent runs. Okay, and not just that, but this instrument also helps you recover your antibodies. So if you wish to actually recover your antibodies because you want to reuse them subsequently, this instrument actually gives users the option of reusing these antibodies. So you can actually reuse them because some primary antibodies can be pretty expensive. So you do have the option of reusing them, okay, with this instrument. Very simple software as well that you can actually program to match your conventional manual protocol. So if you're doing all those steps manually, you can actually just program the steps in this instrument. Okay, so any manual method that you are currently using can be easily transferable onto the EasyWest device. And importantly, it's also an open platform. So what this means is that you do not need to actually purchase any proprietary reagents from GenScript. You can just use whatever you are currently already using manually to load it onto this instrument and use it. So you do not need any additional consumables from us. Okay, so there are no additional consumables costs involved. So this is again just some of the features of this instrument. You can actually probe one set of antibodies at one go, 2 membranes at the same time. And if you have strips of membranes, you can also load them onto the cassette to actually blot them. And it requires a minimum antibody volume of about 10 mils, which is similar to probably what you would use manually as well. It gives you the option of recovering your antibodies, and there is, again, no additional consumables required every time you choose to use this instrument. Okay. So how it actually works is that it's a very small device like this, and you basically put all your reagents at the side here. So on the right-hand side of the instrument, you can open it up, and then that's where you would place your primary antibody, your secondary antibody blocking buffer at the side here. And in front over here, this is an opening where you can actually open this door. And inside the door, you can actually fit a cassette. Okay, so what you put in the cassette would be your membrane that you wish to actually blot. Okay, so you open up the cassette, and then you will see that there are some markings on the cassette itself. So you just place your membrane on the cassette according to the markings, and then close back up the cassette. Okay, and then put this cassette with the membrane inside on the instrument here, okay, and then close back the lid over. Okay, and then just program your method on this software. Again, very easy to use software. Just tell it, how long do you want to perform the incubation for? How long do you want to perform the washes for? And then press start, and then it will automate the entire blocking, incubation, and wash steps for you. Okay. In terms of results, I would say the results are as good as conventional methods. So if you were to do it manually compared to if you were to do it on the EasyWest Lite instrument, you can see that you get very similar results. Okay, very similar whether you do it conventionally, manually, or with the EasyWest Lite instrument, you get very similar results. But of course, the main advantage is that everything is automated for you. So you do not need to perform this spend many hours on the bench doing this. Everything is just done within the EasyWest Lite system. Okay. We also did let customers try our EasyWest system, and as you can see, in fact, this customer actually got better results using the EasyWest Lite system compared to doing it manually. You can actually see an additional band much more clearly using the EasyWest Lite system, okay, compared to performing it manually. This was another test that was done at another customer's place. You can see also you get much sharper or stronger bands associated with the EasyWest compared to performing it manually as well. Okay, but again, conservatively, I would say that EasyWest Lite likely is going to be able to at least give very comparable results compared to if you were to do it manually. Okay. So just a summary again of GenScript's EasyWest Lite device. It's basically an automated device that is used to perform that blocking, antibody incubation, and all wash steps. And the results are as good as conventional methods, the manual methods, but of course it allows you to save a lot of time at the bench. Okay, improves consistency as well because it's an automated system, and you can actually save methods onto the software as well. It's an open platform, so you can actually use commonly used reagents. You do not need to purchase anything from us in order to run the instrument. It's a very easy to use, user-friendly system as well. Okay, so again, do feel free to reach out to us if you wish to have a look at how this instrument actually works, or even if you wish to try running your current protocol on this, we are more than happy to actually let you try it out on our EasyWest Lite device. Okay, so before I end my session, just an overview of the stuff that we've actually talked about for today. Okay, so we've actually gone through the entire, or more or less, most of the Western Blot workflow. Okay, so starting from gel electrophoresis, again, this is for you to separate your proteins on the gel. Okay, and then you perform a protein transfer to actually transfer all the proteins from the gels to the membrane, and then finally you perform that blocking and antibody incubation steps over here, which is again typically done manually. Okay, and at GenScript, we do have solutions again for you if you wish to actually perform any one of these steps or all of them. In terms of gel electrophoresis, we do have our SurePage precast gels, which are Bistris gels, so again more stable. And not just that, we also will provide free running buffer for you if you actually decide to use our precast gels. And we also supply things like gel tanks, protein standards, LDS sample buffer that you can use for gel electrophoresis. And not just that, so once you've performed your electrophoresis, if you wish to perform a gel staining, we do have that e-stain system, okay, that you can actually use to perform your staining, to view the protein bands on the gel. And this is able to complete your staining just within 10 minutes. We have the e-blot system if you wish to perform that gel to membrane transfer. And this, it actually gives you extremely good transfer efficiencies, similar to a wet transfer method, but with the transfer time as fast as a semi-dry. Okay. And lastly, we have the Easy West Light that can help you automate the entire blocking and antibody incubation steps. Okay. So this is the full range of solutions that we actually have at GenScript. And all these can work together to help you reduce your benchtop as well as experimental time with either automation or with ready consumables like our showpage gels, as well as improved technology like the e-stain and the e-blot systems. Okay. So again, these are just some of the things that we have, like that I've mentioned. We have the showpage gels for you to perform that gel electrophoresis. If you wish to perform staining, we have the e-stain system. To perform transfer, we have the e-blot protein transfer system. And for washing and incubation, as well as blocking, we have the Easy West automated device. Okay. So again, do feel free to reach out to us for a demo on any one of these e-devices that we have. And we can actually provide you, we'll actually provide you free gels and protein standards from us as well. So that you can actually use these consumables to run on these devices, and you can actually see for yourself how well they actually work. Okay, so do feel free to reach out to us if you want to give any one of these things a try. Okay, so with that, thank you so much for your time, and I've come to the end of my presentation. So
Hey,
if
Jane?
you guys have any
Yes.
questions, yeah, let me know. Thank you.
Hey, hi, thanks, Jane, for your detailed presentations on the Western Blot workflow. So now we have passed the 1 hour mark, but we can quickly answer some questions from the floor, right? So okay, the first question is: are the showpage gels compatible with the gel tanks from competitors? I mean, from other vendors?
Yes. So, for example, we have actually run many of the showpage gels on, for example, BioRed gel tanks. So you may have to do just a slight modification. You do not need to purchase anything, but you just need to flip the gasket from the current position to the opposite direction, which we can actually show you how to do it. But it's basically just taking out the gasket and then just flipping it onto the other side, and then you can use it with our gels. So it's shown to work very well. So you do not need to purchase anything extra to actually use on the BioRed gels. On some of the thermal gel tanks, you may require an adapter, but don't worry, because we will supply the adapter to you for free as well. So if you find that you're actually using, for example, a thermal gel tank, we can actually provide you an adapter, which you just need to add in that, and you can actually use our gels on the thermal gel tank. So no problems if you have any of the other gel tanks in your labs. You should be able to use it.
All right. So may I use our own staining reagents on this e-stain system?
Okay, so we actually recommend that you use the staining reagents coming from us because the staining reagents will need to be of a certain concentration, because if it's of a different concentration, it may register as an error on the system, on the e-stain system. Okay, so because we do have some sensors within the e-stain, just to make sure that the reagents that goes in is of the correct concentration. So if you actually were to use your own staining reagents, it may not really be compatible with the e-stain system. So we highly recommend that you actually use the staining reagents coming from GenScript, which is also very cost effective actually. Yep.
Yeah, great. We'll answer one last question, right? May I use GenScript gels on the e-blot, and am I able to use the other gels on it?
Yes. So the e-blot is highly compatible with any gels, whether it's homemade gel, GenScript gels, or other brands of gels, it's highly compatible. So as long as it's a mini gel, it should be able to fit onto the eBlot system. Yes.
Yeah, okay, great. So due to the time constraints, we will end our presentations here. So if you have any other questions, right, you can just leave it in the Q&A box and we will answer you by email after the presentations.
Okay. Okay. Thank you everyone for your time.